I hope everyone is doing well today. Welcome and thank you for joining us for today's webinar, Understanding Insecticide Resistance. My name is Chris Walker, Senior Program Analyst here at the National Environmental Health Association, and I will be your host for today. Today's webinar will be the first of a two-part series. Part one is Basic Principles of Resistance Development and Detection Before Product Failure. The second part will be held in 2025, and we will share more information on that upcoming webinar towards the end of this webinar. To begin with today's webinar, I will highlight a few things, one being a partnership between two organizations, briefly providing an overview of each and what they do. I'll next introduce today's speaker, and this person has an amazing presentation planned for you today. And then towards the end, we'll have a Q&A session following the presentation. So an opportunity to ask questions. As we come to the end, we will provide a few final announcements and some upcoming opportunities. Now, a few housekeeping items as we get started. Please note that this webinar is being recorded. The recording will be uploaded onto the NEHA Vectors and Pests webinars page. You can also find recordings of previous webinars there as well. The web address is www.neha.org/vector-webinars. If you have any questions for our speaker during the presentation, please use the Q&A box to submit them, and we will attend to them after the presentation. You can find the Q&A box at the bottom of your screen, so make sure to be mindful of that. At the conclusion of today's webinar, you will receive a link to take part in an evaluation survey to provide feedback and comments on today's webinar. We ask that you take the time to let us know how we can continue to meet your needs as an organization. This webinar is brought to you in part through a collaboration between Gulf South Vector and NEHA through grant funding from the Centers of Disease Control and Prevention. Allow me to share some information about both organizations. The mission at the Gulf South Vector Educational Center for Training, Outreach, and Resources, or Vector for short, is to forge a robust, innovative, and multidisciplinary and cooperative regional network. They aim to provide innovative training and resources to professionals and pre-service students, creating a unified workforce that enhances regional capacity to monitor and respond to vector-borne disease threats. If you want to know more about this organization, you can either scan the QR code that's on the screen or visit their webpage at vectoroutreach.org. Now, for those who may not be familiar with NEHA, we support the environmental public health workforce. As an association, everything we do is to build, sustain, and empower environmental public health professionals so that they're equipped and prepared to protect health of communities across the country. We support environmental public health by continuously providing the most up-to-date training, tools, and resources to the workforce who specialize in so many different fields considered in environmental public health. NEHA is focused on increasing knowledge and skills across all of our fields to build upon our workforce capacity effectively. We also offer credentials to certify the expertise of our workforce. In addition to our large online library of trainings, webinars, and available tools and resources, we also host an annual educational conference, which includes multiple sessions focused on many different fields in environmental public health, such as vectors, food safety, private wells, water quality, climate change, and so many more. No matter where you are in your educational journey or professional career, NEHA has something for you. Please check out our website at www.neha.org/membership. I do want to point out that if you are a student, Currently, membership is free, so please be mindful of that. As I mentioned, each year, NEHA hosts our annual educational conference and exhibition. Our 2025 conference will be in Phoenix, Arizona on July 14th through the 17th of 2025, and promises to be your oasis in the desert. Registration is currently open, and we highly encourage you to attend. You can learn more and register at neha.org/AEC or scan the QR code here on the slide. Now, let's get to our guest speaker. Please welcome Janet McAllister. Janet is a board-certified medical entomologist who earned her undergraduate and master degrees from Louisiana State University. Her PhD in medical entomology comes from the University of Arkansas. After 20 years of public service as a research entomologist with the Centers for Disease Control and Prevention, Division of Vector-Borne Diseases, she is now the principal research entomologist for the City of New Orleans Mosquito, Termite, and Rodent Control Board, where she supervises the Mosquito Control Unit. She is an American Mosquito Control Association Medal of Honor recipient for outstanding contributions to the science and control of mosquitoes, other vectors, and vector-borne diseases. Janet, the floor is yours.
Okay, thank you. Just give me a second to share my screen here. All right, it looks like my screen is being shared. So today I'm going to talk just about some of the basic principles of resistance development and some of the testing methods. And as Chris said, the second part of the webinar will go more into the details of how resistance works and strategies for managing for insecticide resistance. So to get started, where does resistance come from? So if you were to spray a population of insect, and this works for all insects, whether it's fleas, cockroaches, corn earworm, in most of these examples, I'm going to use mosquitoes because that's what I'm most familiar with. But this goes more broadly beyond just mosquitoes. So when you spray a population of mosquitoes or some insect, there will be, just because of genetic diversity, perhaps some individuals that are able to survive that insecticide treatment. And so while most of the population is going to be killed by your treatment, those individuals are going to survive, and they are going to contribute genes to the next generation. So the offspring of that survivor may have the right combination of genes that make them able to survive future exposure to insecticide, and some of the offspring may not have the genetic combination of alleles. But the bottom line is that as you continue to pressure that insect population with insecticide, the ones that have that something special that allows them to survive are going to continue to survive, so that over time, the proportion of the population that has these resistance mechanism genes are going to become dominant in the population until you reach a point where when you spray, nothing is going to be killed. And so that is the process of developing insecticide resistance. Now, there are a lot of different definitions for insecticide resistance, and the traditional definition, which is really used in a lot of toxicology classes, and used by a group called IRAC, which is the Insecticide Resistance Action Committee. It describes the ability of these insects to survive actually multiple normally lethal doses. So you spray with the product and the insects survive, and you spray with the product again and the insects survive, and you spray with the product again and they survive. And the reason that they are surviving is because they have some genetic makeup in the population that is allowing them to survive. That is what they consider insecticide resistance to be. And it needs to be tied to genetic mutations or overexpression of enzymes, because there are things that can make it look like resistance when it's not. So you go in and you spray, and perhaps you didn't pay attention to the expiration date of your product. And the reason why you're spraying and they're not dying is not because they have resistance. It's because you are using a product that may not be effective, or that you are not reaching the insects themselves. But if you want to manage for insecticide resistance and you want to maintain susceptibility or regain susceptibility in a population, then the time to act is before resistance reaches the level where products are not working. And so the resistance definition that CDC uses, and the one that I like, is that it's a genetic change in this population. So it's actually, they are exposed to the insecticide, but they are able to overcome it, and that it may impair control in the field. So this allows you to do that resistance management before you outright lose the use of your products. So some additional definitions are mode of action, and this refers to the way in which that insecticide works. So all insecticides, when they enter into an insect, there is a specific target site within that insect where they will affect some change that is lethal to the insect. And so there's several listed there, and I'll talk more about those in part 2. I also want to define resistance mechanisms. So this is actually a description of what that genetic change is that allows it to overcome those insecticide exposures. And those really fall into 4 broad groups. So you can have increases in metabolic insects that will break down the insecticide. You can have mutations at the target sites of the insecticides. You can have reduced penetration, where the insecticide trickles into the body of the insect because the exoskeleton and cuticle have become thickened or changed so that that insecticide cannot be easily absorbed. And then you can have behavioral changes. And I think the best example of that is resistance in anophelines. In South America, they were doing hut spraying, and they would spray the walls of the hut because the anopheline vector for malaria there would rest vertically on walls. Well, they changed the behavior so that the preferential resting of the anopheline became horizontal, and they would go up to the ceiling and rest. So those anophelines that preferred to rest that way because their genes told them they wanted to be horizontal, not vertical, actually selected for resistance. And they were able to overcome it quite easily by simply treating the ceilings as well as the walls. So the last 2 definitions I want to cover are cross resistance, and this is where you have a common detoxification system or target site, common resistance mechanism that works on multiple insecticides generally in the same class. And so classic example of this would be knockdown resistance, or KDR, for pyrethroids, where it develops because you're using one pyrethroid. And if you come along with a second pyrethroid, that second pyrethroid already has cross resistance to it, even though your population has not necessarily ever been exposed to that second pyrethroid. And that's a little bit different from multiple resistance, because that really, if you think about it, there's a variety of classes of insecticide out there, and there can be a variety of modes of action, and there can be a variety of resistance mechanisms that are in the same population, so that you have resistance to multiple insecticides, but the mechanism behind it is not one mechanism, like in cross resistance, but multiple mechanisms. So you can have the altered KDR, target site resistance, coupled with increases in enzymes, and so you have resistance to both perhaps pyrethroids and organophosphates in the same individual. So the question with these definitions is, at what point do you have a resistant population? And with the traditional definition, where insects are surviving multiple exposures to a product, the bottom right cartoon there is where that definition would say, okay, we have resistance because we are not controlling our population with our product. Whereas the more proactive definition, the point at which you have resistance is going to be much earlier, where perhaps you have maybe just a few, and you want to be very proactive in managing for resistance, or maybe a little bit more when it comes to mosquitoes. WHO actually has some cutoff points for what is a resistant population, and that is when less than 97% of the population is being killed by the insecticide. So they have a pretty high threshold when you're doing insecticide resistance testing. So how do you develop insecticide resistance? If you're using a single class of insecticides, say you only use pyrethroids, and you are using pyrethroids that have long residual action, so they are out in the environment for a very long time, a lot of potential for exposure of the insect, slow release formulations, and you're applying those insecticides to all life states and all generations. So both larvae and adults are being exposed. Every single generation, there's no relief from the pressure of the insecticide, and you're treating all of the habitat, so the insects can't really get away from the insecticide. Then you are more likely to have that resistance develop. Now, that often is why resistance develops very quickly in some of our public health pests, such as cockroaches, because all of those things I just went through apply to classic cockroach control, where you would go in and spray baseboards with a, say, a pyrethroid or organophosphate. Nowadays, there are a lot of baits, so that not all generations and all life stages may actually be exposed to the insecticide, which helps to prolong the use of the insecticide. When it comes to mosquitoes, we don't use the same class of insecticide when we're trying to reach the larvae and pupae in water versus the adult mosquitoes, which are terrestrial. And then for our adulticide space spraying in vector control, we're not using long residual action products or slow release formulations. Those actually tend to be more used toward the larval stage. And there are a lot of factors that influence the selection of insecticide resistance. So one of those are the genes of the population or individuals in the population. So what's the frequency of resistance allele? How many different alleles are present to contribute to that resistance? Are they dominant or recessive? There's also biological factors that affect whether a particular insect species is going to develop resistance or not. How many generations? How quickly do they reproduce? What are they eating? How isolated or mobile the population is. If it's something like an example for mosquitoes again is it a species that the adult mosquito can fly 25 miles in a night, or is it a species that is confined to 150 meters of movement during its entire lifespan? What they eat, and then whether there is some sort of refugia, those are all biological factors. And then the third thing that can influence insecticide resistance is the operational side, and that is what chemicals are we choosing? What relationship do those chemicals have to prior chemicals that have been used? Are we using these long-lasting formulations? How are we applying them? What kind of application methods? What life stages are we targeting? And when it comes to managing for insecticide resistance, that is the only category of influencers that we really have control over. We're not, at this point, we can't, move genes in and out of populations. We're not going to change the basic biology of our insects that we're trying to control. So our option is our selection of chemicals, our selection of strategies, and how we apply insecticides. So besides just the chemicals that we pick, there are some other influencers of whether resistance is going to develop or not. Because even though an herbicide is meant to kill a plant, it may, if it runs off into water, or if it's an aquatic herbicide, it's going to influence other insects that are in that water as well, because it may cause them to need to have additional enzymes to be able to survive habitats that are more highly polluted. And so fungicides, fertilizers, just non-point source runoff can have a huge impact in some of our mosquito species, and whether they are more likely or less likely to develop insecticide resistance. And then where do our chemicals come from? And so it's not just vector control that is out there trying to address the control of our vectors. Urban pest control, those PMPs out there are applying insecticides. Agriculture can have an influence. And then the do-it-yourself market, the homeowners, the businesses that do spraying on their own, often without much education on what it is they're doing. And so all of those things can influence whether resistance is going to develop or not. So I want to say something about insecticide resistance surveillance, and just really define what I mean when I say surveillance. So a survey is really a one-time gathering of data to assess a situation. And that is different than surveillance, which is this continuous process of monitoring changes over time so that you can make decisions on when you might want to adjust your control measures. And so what does that look like? It really, for resistant surveillance, it looks like a series of snapshots. So you do some testing, you look at your population. The first time you do it, it's a survey. If you never do it again, it's a survey. But what you should be doing in your program is surveillance. And so you should be taking new snapshots, and hopefully your new snapshot looks like your first snapshot. And you may see your new snapshot look maybe a little bit different than your initial snapshot. So that's telling you two things. One, it could just be that when you did your testing, you get a little bit of variability in your results, or it could be that you are actually starting to select for insecticide resistance. What you don't want to see is that your new snapshot doesn't go back to looking like your initial snapshot, but that it goes from maybe a little bit different to a lot different to a lot different, because that tells you you are definitely selecting for insecticide resistance, and you need to make some sort of change in your program to try and maintain susceptibility of your population. So when it comes to testing, there are a lot of types of resistance assays out there. And I will say that laboratory assays, even if you're using a formulated product and making dilutions, does not correlate with operational control parameters. So doses and assays do not equal label rates on products. And there are multiple ways to deliver the insecticide when you are doing these assays. So you could be directly applying the insecticide to the insect using syringes. We often call that topicals. You could be coating petri dishes or bottles. You could be dipping leaves into insecticide and then having the insect come in contact with the leaves. There's a lot of different ways that these assays can be run, but they fall into really three different groups of assay. And so the first one I'll talk about is the time mortality assay. CDC has designed one of these, this assay, and they actually have a program where if you're a mosquito control program or public health program that does mosquito control, you can actually get these kits for free and do this assay on your own. But the chief advantage of the time mortality assay is that it is measuring a direct critical toxicological parameter, and it is the length of time that it takes for a set dose of insecticide to reach the target site within the insect in the absence or presence of resistance mechanisms. And so this is a little bit different than the assays that I'll talk about next, which are dose-based assays, because those dose-based assays measure a relative change. So you are doing a comparison of a dose against a dose that kills your test population versus a dose that kills a laboratory or susceptible population. So what does this look like? If you're looking at an insect represented by the blue rectangle and you expose that insect to an insecticide, which is represented by the circle with the little black dots in it, those little black dots are molecules of the insecticide, and they penetrate into the insect. And along the way, on their way to the target, some excretion is going to naturally occur. Insects have enzymes that are similar to the enzymes in our livers. So our livers detoxify things that we ingest. Same thing happens with insects, even though they don't have a discrete organ called a liver. And then that insecticide gets to the target site and interacts with it. And that is a discrete time that it takes for this to occur in an insect population. And so if you're using the same dose, and you can set a time that it will, you can select a dose that allows you to select a time that is discrete, and therefore, when you test populations in the field, you may be able, or you will be able to see a change in that time. And that's really what is represented here is the susceptible insect. You have a specific dose, you have a threshold time that you know it's going to take those susceptible insects to die. But in a resistant insect, you can have reduced penetration that slows down the insecticide getting to the target site. You can have increases in excretion and increases in detoxification that make what normally would be a lethal dose a non-lethal dose. And then you can also have modifications of the target site. And the net result in the bottle bioassay or a time mortality assay is that you measure the increased time that it takes for that insect to be overcome by the insecticide if it dies at all. So the data generated from bottle bioassays, again, we select a discrete dose and we use susceptible mosquitoes to determine a resistance threshold or a time in which we expect 100% of those susceptible mosquitoes, or if you were to do this with other insects, I've worked with fleas on this assay as well. You have your resistance threshold and you expect 100% mortality at a specific time. So the data coming out of a bottle, if you have a resistant population, all of the mosquitoes that are dying on the left side of that threshold time are individuals in the population that have susceptible genes and they're being killed by the insecticide. Whereas all of the mosquitoes or fleas, or if you were to do this with cockroaches, all of those insects to the right are going to be the proportion of the population that has resistance being expressed in it. So your first data point of interest is that proportion of the population that is being killed at your threshold time. But this assay strategy also gives you some important information about how strong that resistance mechanism is. And so the shape of the curve, especially the shape of the curve past the threshold time, is important. So you can have two populations of mosquitoes. One of them, all of the insects, they have roughly the same level of individuals or proportion of the population that is surviving beyond the threshold time. But in one population, they may eventually succumb and all die a couple of time points after your threshold time, versus a population that even after 2 solid hours of being exposed to the insecticide, they're still not dying. And so that gives you an indication of the strength of the resistance mechanism in that population. And certainly it would be much more troubling to see a population that is equivalent to that resistant or purple line versus the light blue, because with the light blue, it's going to be much easier to manage to get susceptibility back into that population. So the time it takes for that insecticide to work, it is dependent on the dose, and we set a discrete dose, but that also varies by species. And so if we set a discrete dose that varies by species, what is varying by species is actually the time that we're able to measure. And so one species of mosquito in that CDC bottle bioassay may have a threshold time of, say, 45 minutes, and another species using the same chemical at the same dose is going to have a threshold time, maybe 60 minutes. But the important point is that the intrinsic susceptibility to toxicants is variable depending on the species itself. And then if you are someone who is going to do resistance testing and you want to publish data, the appropriate statistics to use to analyze time mortality data is either a Weibull distribution or a Kaplan-Meier test. And I point this out because those are not the traditional tests that would be used in dose mortality type assays. So I'm going to switch and talk a little bit about dose mortality assays. They measure a relative difference in doses that it takes to kill an insect population. And so usually it's an indirect measure because you need this to determine the LD50 of a susceptible or colony population. And then you run the same test on field populations and you determine the LD50 of those field populations. And then generally you're going to do a resistance ratio or a ratio of what the LD50 for that field population is compared to a colony. And so the little example in the bottom is how a topical assay might be done, where you knock out your insect, or if it's slow moving, maybe you don't need to knock them out. You would weigh them so that you can get an accurate dosage of amount of insecticide per body weight. You would then treat them with the insecticide, and then you would hold them for some amount of time. Often it's 12 or 24 hours, and then you count the mortality. And you do this with multiple doses so that you can generate a mortality curve. And the question is, when is the population significantly different from your colony population to call it resistant? So when you are doing a dose mortality assay, generally you use what's called a probit analysis approach. And so you will use a series of dilutions, and then you will graph that with series of doses and graph that against the percent mortality at each dose. Now, one of the things I pointed out for the time mortality is that the shape of the curve is important. In a probit analysis, the slope of your curve is important. And so that tells you how much genetic variability you have in your population. So if your line is like A, where it's more vertical, you have a much more homogeneous population than if your slope of your line, it looks like B, where so it's just a showing you how much genetic variability you have. Now, if you're doing this dose mortality and looking forward to manage for your resistance, it is better to have more genetic variability, because that means you have more individuals that have susceptibility genes to allow you to manage and regain susceptibility to a particular insecticide. And there are some assumptions using a probit analysis, and that you have a normal distribution of your population. You have a large sample size to be able to detect differences, and then your data should not be bimodal. So what I mean by that is a normal distribution is the bell curve. A bimodal distribution has two lumps in it instead of one bell. It may have two bells. And then the other thing is the mortality between time points is independent. And that's important because if you try to take probit analysis and use it on time mortality data, you violated one of the basic assumptions for even using that statistical test, because in a time mortality assay, the number of mosquitoes that are dead at 60 minutes is dependent on the number that were dead at 45 minutes. So they don't undie as the test goes on. Whereas in a dose mortality assay, the mortality between the doses is independent. So if you're going to report dose mortality data or probit data or a logistic regression, most journals have a standard minimum amount of information you need to report: how many were test subjects you had, your slope, your standard error, what your LD or LC, which stands for lethal concentration, is with confidence intervals. Chi-square, so this goodness of fit, if you don't have a good chi-square, that's an indication you have a bimodal population and that your probit model does not fit the actual data that you have, or that the doses you selected to run in the test don't cover the range of variability that you see in your population. And then there should be some sort of statistical test that you run to show what the best model is, whether you should actually be using a probit test versus a logistic regression. So an offshoot of dose mortality testing is what's called discriminating dose assays, or a discriminating dose assay. And rather than doing multiple doses of the insecticide and generating a probit line, you find a discriminating dose that you are going to use to test against. So you just have one dose in the assay and you test your field population and you record the percentage of mosquitoes or fleas or corn earworms against that discriminating dose. And it is usually expressed as the proportion of the insects that survive that discriminating dose. And again, this is often expressed as a resistance ratio as well, where you would compare it back to maybe a susceptible population, or that you would compare different populations using a ratio. This does require dose mortality assays to determine what that dosage is going to be. So there is a lot of work on the front end to figure that out. So when would you use that dose mortality versus a discriminating dose? Usually a dose mortality is going to be used if a small change in your population is suspected. So it's dose mortality assays are probably the most sensitive resistance assays out there. But to do a dose mortality assay properly, there's range finding that has to be done. So that dose mortality assay, as I said, uses multiple doses that you expose your insect to, but what is the range of those doses? What's the lowest dose versus the highest dose? It's not just I'm going to use a 10 times and 100 times and a 1,000 time difference in my doses. You would do that maybe in your range finding, but you need to find a range that is going to kill very few individuals at the lowest dose and almost 100% of the individuals at the highest dose. And that's going to vary by species of insect that you are looking at. Discriminating doses, on the other hand, can be used if you're looking for a very large change in the response to that discriminating dose in a small portion of the population. And just a comment on the insects you use for resistance testing in general. Oh, there was one other thing I did want to say about, go back. When you're doing a discriminating dose assay, the dose that you're going to choose should kill 100% of a susceptible population, but kill very few to no insects in a homogeneous resistant population. So often trying to set that discriminating dose is difficult because you have to have both a known resistant population and a known susceptible population to create a good discriminating dose. So getting back to the, if possible, when you are dealing with testing for insecticide resistance, it is best to choose individuals that are as close to possible as you can get to field collected mosquitoes directly, or fleas or whatever. Now, sometimes that's not possible, and you might have to raise one generation, an F1 generation, to get enough individuals to do the test. But if you are going to an F2 or beyond, every time you raise a new generation in the laboratory, you are moving the genetic variability of that field population further away from what is actually out there that you are trying to kill. You need to maintain colony and field insects under the same conditions. So they have to be reared side by side in the exact same manner. And so you're not going to rear your colony, say mosquitoes, at a different temperature than you would be holding mosquitoes that you collected from the field. And you also want to know, or at least try to manage so that the age of the individuals in your assay, both the age of the mosquitoes or the age of the fleas in your colony versus the field are the same, but also the age of the individuals are all the same. You want to control for sex. So there can be differences in response to an insecticide based on whether it's a male or a female. The physiologic state, have they been fed? Have they been blood fed? Have they been sugar fed? Are they oviparous? Do they have eggs developing in them? All of those things are going to affect your test results. And so you need to try and control for all of those things when you are doing resistance testing.
Janet,
And
let you know you have about 2 minutes.
okay, well, that's probably good because third to last slide. So when it comes to surveillance for resistance, whatever test that you choose to use, whether it is dose mortality, discriminating dose, time mortality, if you do some sort of field test, it's all about surveillance, detecting what's in your population, making a choice. Am I going to continue to use the same chemical in the same manner, or do I need to change it based on the results of my testing? And then evaluating, did that change make a difference? Was I able to control my insects? And then continuing to do the testing again and seeing, do I need to again adjust my control actions or not? And so it's kind of this continuous loop of detection, taking an action, and evaluating the effect of that action. And I can't stress enough, most people think once you have resistance, that's it, and that product will no longer work. If you use the more proactive definition of resistance and you make changes when you detect a difference in your population, you can preserve the use of products, and you can also, if you catch it early enough, you can reverse the resistance in a population and have regained the use of insecticides that were once expressing resistance. So for day-to-day surveillance, use an easy lab assay, something that'll work with you, monitor throughout your area of concern, whether it's an entire county or whether it's just a town or a city. When you detect resistance, you probably want to do some additional exploring to confirm its impact on control. Change your control strategy and look, because populations can recover. So continue to do that surveillance if your population has recovered, and you might be able to resume your original control strategy. Maybe not, maybe you don't want to. And then how long does all of this take? Well, the only way you're going to figure that out is to do day-to-day surveillance for resistance. Now, I don't literally mean day-to-day. Most people can't go test their mosquitoes every single day, but the interval of time should be appropriate to the biology of the insect. So if you have a very short-lived, very reproductive insect, you may want to do your snapshot or your testing more frequently than if you have something that is longer lived. Our suggestion when I was with CDC was to do a minimum of once a year. And then if you had the ability to try and do pre-season testing and post-season testing, but that's not going to apply to all public health insects. So with that, there is my contact information. If you want to reach out, please feel free and open it up to questions.
All right. Thank you, Janet, for the presentation. We have time for a few questions. So first question to you, Janet. Are certain mosquito species more likely to develop resistance to insecticides than others?
Absolutely. Absolutely. So a species like Culex quincunfasciatus, which is our primary West Nile virus and St. Louis encephalitis virus, the biology of that mosquito is that it prefers to put its eggs in nasty, polluted water because there's generally no predators that can survive in that water. And so for their larvae to be able to develop, they have to be pretty robust. They have to have just hardwired into them some pretty robust detoxification enzymes. And so they are more likely, because they come preloaded with lots of these enzymes, to develop insecticide resistance versus a mosquito species that lives in cleaner water, such as a lot of the anophelines in the US, where we do not do residual treatments. And so we're controlling them in the larval stage. And because they live in such clean water, they're not as likely here in the US to develop resistance. The other thing is our Culex and our Aedes aegypti, they don't go very far. So if we're treating our backyard and they're not going very far, and you're doing residual sprays in your backyard through a pest control company, then they're being exposed much more than a species that you may spray, but they're dispersing miles away from where you may be spraying as well. So it's gets down to that biology part that
Okay.
that makes a difference.
Gotcha. Okay. And this will probably be our last question. How can public awareness and education help reduce the spread of insecticide resistance in mosquito populations?
So I think we're a community where public health doesn't work in a vacuum. We've got pest control operators that also do mosquito control treatments. We have vector control programs. We have homeowners, which are maybe one of our bigger drivers of resistance development. And so educating people that it is a thing, and to use their insecticides according to the label and don't overuse them, can go a long way in helping to prevent insecticide resistance from developing. So it's a community thing. It's not just us, but generally it's mosquito control programs or public health programs that do mosquito control that would have the bandwidth to actually test. And so I would encourage anybody who does resistance testing in a mosquito control situation, um, to to share that information, so that if you're seeing an issue, it's not being compounded by what others are doing.
Got it. Thank you for that. Unfortunately, we are at our time. I do see a question in the chat. We're going to send the recording via email. So we'll do a follow-up communication for the question, and we'll probably have that comment or that question, a response for that one. So I do want to take time just to thank Janet McAllister for the presentation. This was extremely helpful. So we're now going to let you know that, as you can see, we do have our next webinar that's going to be in January. So you see that information, feel free to take the opportunity to register. We have the link that's available in the chat. That's going to be on the second part, which is mechanisms and management theories. So if you enjoyed this webinar, we invite you to come back for the next one, which is going to be in January. So please be on the lookout for that one. And as we close, we just thank you for your time. We hope you consider being a member of NEHA if you are not also. The more members we get, the stronger our voice can be to address some of these issues on a national level for policy and funding. So thank you all for your time. Thank you, Janet McAllister, for your presentation. Thank you for joining us and wishing you all a happy holidays and hope to see you again in 2025. So take care. Thanks a lot.
Thank you.
Thanks, Janet.